I will start by introducing different types of high-dimensional data. A common one is text documents. So there's software that can identify plagiarism. Or there's other software that are email runs to identify junk email and filter it from your inbox. Images provide another data type. Facial recognition software can be used to unlock smartphones and computers. And in the health sciences field, we have examples that include time series of physical activity that's collected through smart devices, as well as spatial data collected in air pollution studies, and molecular genetics. And these are all collected to help us better understand human health. So the data I'll be using in the class for teaching statistical analysis will be from molecular genetics. This figure is taken from the ENCODE project's website. ENCODE stands for the Encyclopedia of DNA Elements, and it's provides data from a large collaboration that started back in 2003. The picture captures the complex molecular structure of DNA, and the blue boxes below show an assortment of different technologies that are available for measuring the different molecular structures. Each of these technologies generates data that's genome-wide and would provide a high-dimensional data set. So we're going to analyze, to start, gene expression data, which is just one of these types. And in the last few weeks of class, I'll discuss methods for combining multiple high-dimensional data sets. So if we think of gene expression, when I say we're going to measure gene expression, the central dogma of molecular genetics is that our DNA is transcribed into the RNA, and that RNA then is translated into proteins. So it's the RNA, this middle step, that we're going to be measuring in our data sets. So common biological questions that we can ask with this type of data are, so what genes are differentially expressed between different groups, where groups are different treatment of cells or different patient sets. That's one type of question. A second question could be, can we diagnose disease subtypes using gene expression? So in leukemia subjects, can we, from the gene expression profile in their blood, identify which subtype of leukemia they have? And then a third question is, can we actually discover novel disease subtypes from their gene expression profiles? So if we have a solid tumor, a solid cancer, can we look and identify subtypes of cancers that are different from one another? So by the end of this class, we'll be able to apply the appropriate statistical method to answer each of these different types of questions. There are two main technologies for measuring gene expression, microarrays and sequencing machines. So data from both methods will be analyzed in the class. The main difference is that the arrays are designed with a probe, or lots of probes, attached to the surface. Each probe is designed to measure a specific sequence recognized from a particular RNA product. So these probes on the array can measure on this array over 47,000 sequences. With sequencing, you have flexibility in how you summarize the count the sequences, because you actually get the sequences themselves. So I'll just sort of contrast them quickly here, saying that the microarrays do have a limitation, that you had to know what the 50 base pairs look like in advance, and it was designed on that array. That can lead to limited ability of detecting, say, a splice variant of a gene that might be too much similarity. So if there's too much similarity between different sequences, it cannot distinguish them well. It's just kind of a property of how the data are measured. They also, unfortunately, their lower detection limit is not super low, so their genes that are expressed at low levels might be below that detection limit. And those issues are solved basically with sequencing. So with sequence data, you can summarize the data however you like, whether it be for the whole gene, or you look at a specific transcript, or focusing on a given exon. There's also, because you've got the sequences, you have the ability to discover new alternate splicing that wasn't known when the arrays were made. And then there's a wider dynamic range too, so you can detect lower levels of expression by sequencing more deeply. So there are lots of advantages to RNA-seq, and it's really what I think people are doing today. But initially, it was much too expensive to do RNA-seq, so it was just used to be cost effective to do microarrays, but that's kind of been taken out of, no longer the case. But ultimately, regardless of which way we've collected the data, we're going to create a data matrix that looks like this. So the techniques we're learning aren't specific to either of those two data types. They'll be for a rectangular matrix, we'll say, and we call this, as statisticians, they use the language large p, small n. So p is measuring the number of features. You have many thousands of features measured on each of your individual samples, and the number of samples is going to be much smaller than the thousands of features. So it's, again, we can write it like this, where the number of features, p, is much larger than the number of samples in our data. And you may notice this matrix actually may look flipped to you or transposed from how one typically arranges their data in statistical analyses, where you have your samples on this axis and these are the features. But this has been the standard from early on in gene expression. So some of the software actually expects the data in this format, and we'll talk about that when we go through the different software packages. So here's just, I'm going to end by saying there's a wealth of publicly available data in molecular genetics, and these are a sample of websites and projects that provide data from U.S. government-funded research. And some of the first data set that we're going to download and play with and study with our statistical learning is going to come from Gene Expression Omnibus, and it was a study in which I was involved here at USC.